What will be the learning objectives of today's discussion? We will just recapitulate the subdivisions of respiratory tract, and we will discuss the formation of laryngotracheal diverticulum and tracheoesophageal septum. We will talk about the development of larynx, trachea, and lungs along with their congenital abnormalities. Let's recapitulate the subdivisions of respiratory tree. Functionally, the respiratory tree is divided into conducting part and respiratory part. In the following figure, we can see that conducting part includes upper respiratory tract and the lower respiratory tract up to the terminal bronchioles. Below the terminal bronchioles, we have the respiratory part, which is responsible for the exchange of gases. Coming to the anatomical subdivisions of respiratory tree. Anatomically, the respiratory tree is divided into upper respiratory tract, which includes nostrils, nasopharynx, pharynx, and larynx. Beyond larynx, the respiratory tree is included in the lower respiratory tract, which includes trachea, bronchi, bronchioles, and alveoli. Let's begin with the development of respiratory tract. Which germ layer is responsible for the formation of respiratory tract? It is derived from foregut endoderm. So, our respiratory system is endodermal in origin, and some contribution is also given from the surrounding mesenchyme or the mesoderm, which will form cartilages, muscles, connective tissue of the respiratory system. So, endoderm forms epithelial lining of the trachea, larynx, bronchi, alveoli, and the splanchnic mesoderm forms cartilage, muscle, and connective tissue of respiratory tract and the visceral pleura. So, in figure A, we can see the sagittal section of embryo of 4 weeks, and here we can appreciate yellow color gut tube. We know that gut is divided into foregut, midgut, and hindgut. And from the ventral aspect of foregut, we can see the origin of a small bird-like structure, which is the respiratory diverticulum, and it will form respiratory system in future. In diagram B, we can see further development of this respiratory diverticulum. What marks the beginning of the formation of the respiratory system? A groove develops in the ventral pharyngeal wall just below the hypobranchial eminence in 4th week of intrauterine life. As we can appreciate in figure C, that a groove-like structure is there in the vicinity of 4th and 6th pharyngeal arch just below the hypobranchial eminence. This groove is known as laryngotracheal groove. It deepens to form laryngotracheal tube, and its continuation is the respiratory diverticulum. So after the formation of groove, there are two ridges which develop laterally, longitudinal ridges, and these ridges are known as tracheoesophageal ridges, which we can appreciate in diagram A and B. These tracheoesophageal ridges, they eventually fuse together to form a septum. And why this septum is needed? Obviously, to distinguish the two tubes, the food pipe and the windpipe. Both of them, they have to be separated, and for that purpose there is a development of tracheoesophageal septum, which separates trachea located ventrally from the esophagus, which is located dorsally. So in this diagram we can see a longitudinal tracheoesophageal septum, how it develops and divides the diverticulum, respiratory diverticulum, into a dorsal portion, which is indicated by yellow color in these diagrams. And this dorsal portion will give rise to the esophagus in future and oropharynx, and ventral portion will give rise to larynx, trachea, bronchi and lungs, and it is indicated by blue color. Here we can see the that respiratory diverticulum ends into two buds, which are the lung buds. What is the fate of laryngotracheal tube? Its upper or the cranial part forms larynx and trachea, and the distal lung buds, they form bronchi and lung parenchyma. Let's begin with the formation of larynx. Larynx develop from cranial most part of laryngotracheal tube. What is inlet of larynx? It is the communication between laryngotracheal tube and pharynx. So this communication persists in the adult life as the inlet of larynx, and this inlet is surrounded by mesenchyme of 4th and 6th pharyngeal arches. In the beginning, the shape of inlet is U-shaped. Later on, due to the proliferation of mesenchyme of 4th and 6th arches, inlet becomes T-shaped, and it assumes its adult shape by reorganization of laryngeal cartilages. So, after discussing the inlet of larynx, we will see that the epithelial lining of larynx is endodermal in origin, and the cartilages and muscles of larynx, they arise from mesenchyme of 4th and 6th pharyngeal arches. So, the cartilages are epiglottis, thyroid, cuneiform, cricoid, arytenoid and corniculate cartilages. Out of these cartilages, epiglottis, thyroid and cuneiform are derived from 4th pharyngeal arch, whereas cricoid, arytenoid and corniculate cartilages are derived from 6th pharyngeal arch. When we talk about the interior of larynx, initially what is there? Larynx is a luminal structure, but there is occlusion of lumen of larynx because of proliferation of epithelium. So the lumen gets obliterated. But around 10 weeks of intrauterine life, there are two processes which occur. One is vacuolation and second is recanalization, which produces the recesses, which are known as ventricles of larynx. Endodermal folds are present on either side of this recess. The upper folds are known as vestibular folds, whereas the lower are the vocal folds. And these vocal folds, they develop at the junction of 4th and 6th pharyngeal arches. And this justifies the innervation of larynx, that above the vocal fold, sensory nerve supply, it comes from internal laryngeal branch of vagus, and below the vocal fold, it is supplied by a recurrent laryngeal branch. In these diagrams, we can see at the 4th week of intrauterine life, laryngotracheal groove, it is present and which persists as the inlet of larynx, and that how the various changes are occurring in the shape of inlet of larynx, and ultimately it becomes T-shaped. So, the congenital anomalies of larynx. Laryngeal atresia is the failure of recanalization of larynx, and it results in obstruction of the airways. and it can be detected by ultrasound. Another condition which can be seen is the laryngeal valve. It is because of partial recanalization of the larynx during 10th week. A web is formed at the level of vocal cords and this partially obstructs the airway. Then comes laryngiotosis. It is a condition in which larynx lies low down in the neck. Then there is a condition which is known as laryngocele, in which laryngeal saccule is abnormally large. In some cases, duplication of vocal cords can occur. Let's discuss formation of trachea. As we have seen that cranial most part was forming larynx. Now the intermediate part between the larynx and point of bifurcation of laryngotracheal tube forms trachea. And just like larynx, endoderm forms epithelium of trachea, whereas mesenchyme forms cartilage, connective tissue and muscles of the trachea. Most important clinical condition related to trachea is tracheoesophageal fistula. We already know that fistula is an abnormal tract which develops between two tubular structure. So if there is any defect in the formation of tracheoesophageal septum, it results in the formation of these fistulas. There can be variety of tracheoesophageal fistulas. It is also known as TOF in short. So various type of fistulas can be seen, as we see in figure A, that how the esophagus is separated into two parts, and the proximal blind part of esophagus, it is present, whereas the distal part of esophagus, it has a communication with the trachea. This is the most common variety of tracheoesophageal fistula which is observed clinically, and it is present in about 85% of subjects. In image B, we can see that the esophagus is not communicating with the trachea at all, and both the proximal as well as the distal parts of esophagus are ending blindly. Figure C is the condition in which this type of fistula is known as K fistula, and here both these parts they come together, proximal and the distal part of esophagus, and they open into the trachea. Then D is just opposite of image A. Here the proximal part of esophagus is opening into the trachea and the distal part is ending blindly. E is H-shaped deformity. Here the proximal and the distal part of esophagus both have separate opening into the trachea. Sometimes this tracheoesophageal fistula is associated with other defects also, and here we observe two type of conditions. One is veter and the second is vectoral. T in both these clinical conditions stands for TOF, that is tracheoesophageal fistula. But in veter we have vertebral anomalies, anal atresia, esophageal atresia and renal atresia associated with TOF. And in vectoral deformity we have veter plus cardiac defects and the limb defects. The additional C here stands for cardiac defects and L stands for limb defects. So what is the frequency of occurrence of TOF? It occurs in approximately 1 in 3000 births, and most common type of TOF is the one which is seen in figure A, in which the distal part of the esophagus is opening into the trachea, whereas the proximal part is ending blindly. TOF is associated with prenatal and the postnatal complications. Prenatal complications are polyhydramnios, whereas postnatal complications can be GI associated and respiratory system associated. Surgical repair results in 85% of survival. In this diagram, we can see a fetus, male fetus with trisomy 18, And here we can observe that the upper esophageal segment is ending blindly Next condition is the tracheal agenesis, in which there is trachea has not developed at all and lungs they bud off from esophagus. Then we come to formation of lungs. We have seen that the laryngotracheal tube was ending into two lung buds. So it was bifid. And so lung buds they start developing during 4th week of intrauterine life. In this picture, that this pink color structure is intraembryonic coelom, from where all the body cavities they are derived. And initially the pericardial cavity and the peritoneal cavity they communicate with each other through pericardio-peritoneal canal, and these pericardio-peritoneal canals they are filled with expanding lung buds, as we can see here in this diagram in figure number A. Pericardio-peritoneal canal has been labeled. So this blue color lung bud, it is invaginating into this laterally present pericardio-peritoneal canal. But later on, this pericardio-peritoneal canal, it is covered by folds, pleuropericardial and the pleuroperitoneal folds, which separate the canal from pericardium and the peritoneal cavities. And the expanding lung buds they just punch into this canal, and the mesoderm which is covering the outside of lung develops into visceral pleura, whereas the mesoderm which covers the body wall from inside forms the parietal pleura, and the space between is the pleural cavity. So in second figure we can see that how the lung bud is expanding, and it is just invaginating into the pleura. Visceral pleura is the one which is present immediately around the lung bud, and here we can also see how the pleural cavity it is also decreasing in volume, and the outermost layer is the parietal pleura. So these are the various stages in the developing pleural cavities, that how the pleural pericardio-peritoneal canal they are obliterated by the formation of certain types of folds, and ultimately they form the pleural cavity. So after the we have various stages in the formation of lung. Various subdivisions are seen, that how this trachea divides into two primary bronchus. In figure B we can see that it is dividing into two primary bronchus, right and left. The right primary bronchus it is more in line with the trachea, and it is larger in size as compared to that of left primary bronchus, which is lying transversely. In diagram C, which is the situation around 32 days, we can see the expanding primary bronchus. So and then this primary bronchus or the principal bronchus divides into secondary bronchus or the lobar bronchus around 33 days, and there are 2 secondary bronchus for left lung and 3 secondary bronchus for right lung. And ultimately these secondary bronchus they divide into tertiary bronchi or the segmental bronchi, 10 each for both the lungs. So we can see that how these numerous divisions they are taking place of the respiratory tree, and with the expanding thoracic cavity which is responsible for accommodating the increased volume of lung, the subdivisions they are taking place. Now how many they are there in the formation of lung? There are actually 23 generations of the respiratory tract. And by the end of 6 months of intrauterine life, only 17 generations of subdivisions are formed, whereas 6 more subdivisions they occur during the postnatal life. Now we talk about the uh, further stages in the of lung, and these stages they result in the complete formation of lungs, and they are responsible for the gaseous exchange, which is the main function of our lungs. So there are 4 stages in the maturation of lung. Number 1 stage is pseudo glandular stage, which extends from 5 to 17 weeks of intrauterine life, and this is known as pseudo glandular stage because during this stage the lung looks like a exocrine gland. And bronchi they divide only up to the terminal bronchiole stage, and in this period the respiratory portion of lung is not developed. So if the fetus is born during this stage, it is not viable, that is, it is not capable of survival. Second phase is the canalicular period, which extends from 17 to 26 weeks, and it is characterized by the formation of respiratory bronchioles, alveolar ducts, and few alveolar sacs. Vascularization it also begins during this stage, and there is development of type 1 pneumocytes. Initially, the epithelium of respiratory system is cuboidal, right? And during this stage, it starts becoming squamous, and some cells they become type 1 pneumocytes. And type 2 pneumocytes they also start developing, and they are basically acting as stem cells and are responsible for the production of a phospholipid which is known as surfactant, right? So surfactant production it is beginning and during this period, and limited respiration is possible. If the fetus is born, then intensive care has to be uh, given to the fetus for survival. Then we have secular stage or the terminal sec period, which starts from 26 weeks and it extends till the birth. And in this, more terminal sec they develop, and the epithelium becomes attenuated and thin, and vascularization it becomes rich. Blood air barrier it becomes well developed, and now the surfactant production it increases many folds, and it is sufficient to prevent the collapse of lung or atelectasis. And the last stage is the alveolar period, which starts from late fetal period to age 8. So it extends after birth also, and alveoli like structures are present by 32 weeks. Epithelial lining becomes extremely thin. 95% of the mature alveoli they develop after birth. So the development of alveoli it is continuous and it extends till the 8 years of postnatal life. So here we can see the canalicular period, that how the respiratory bronchiole has developed during this stage. Epithelium is cuboidal and blood capillaries they have started forming. The second stage here is the secular period, which is shown over here. Here the vascularization it becomes more prominent, and some cells they become squamous, and surfactant secretion it also starts taking place. Type 1 squamous cells they are present. And the third is the alveolar period. Here we can see that how the blood air barrier is formed over here, and type 2 cells they become prominent and surfactant production increases. So at birth, alveoli they continue to mature even after birth, and they become more muscular. Growth of lungs after birth is primarily due to the increase of respiratory bronchioles and alveoli. So the respiratory part, it develops basically after birth. And initially what happens that when the neonate is born, the lungs are filled with fluid, and with the first breath, the first breath which the fetus takes, this fluid is squeezed out of the alveoli, and the surfactant, it is present, and this lowers the air blood tension, and that is the reason that the alveoli, they do not collapse during exhalation because of the presence of this surfactant. Now there is an important clinical condition which is seen in case of premature infants, and this is known as respiratory distress syndrome or hyaline membrane disease. What is the cause of this disease? It is the deficiency of surfactant because of the lack of the presence of type 2 pneumocytes. Surfactant is not produced in enough amount, and because of this, the alveoli, they collapse, or there is labored breathing, and there is a deposition of protein in the alveoli or hyaline like structure, which is shown in this picture, in the histological picture of developing lungs. What are the anomalies associated with lungs? We can have congenital cysts, which can be filled with fluid or air, and then there can be the agenesis of lung, which can occur bilaterally or unilaterally due to the failure of the development of respiratory bud. Unilateral lung agenesis is compatible with life. Then there can be inadequate branching of lung, resulting in lung hypoplasia, and there is reduced lung volume. Extreme hypoplasia is not compatible with life. There can be abnormalities of lobes. There can be absence of fissures, which results in reduction in the number of lobes, or there can be additional fissures, which results in extra number of lobes. There is a condition which is known as azygous lobe, in which a part of upper lobe of right lung come to lie medial to the azygous vein, and this part is called azygous. Let's see the development of respiratory system. We can see the development of respiratory diverticulum and formation of tracheoesophageal ridges, then septum, which separates the foregut from developing respiratory system at fourth week of intrauterine life, and at fifth week we can see the development of secondary bronchus. And week 6 we have the development of segmental bronchus or tertiary bronchus, and then we have the development of bronchioles and ultimately the alveoli, which extends even after birth. This is yet another diagram for the development of respiratory system. Thanks.